So the Raas system is important for regulating blood pressure and understanding the path behind hypertension, heart failure, and renal disease. And it's typically controlled by four substances: renin, angiotensin 1, 2, and aldosterone. So these four substances are comprising of a system that responds to stimuli to control blood pressure. The four stimuli, which are typically detected by juxtaglomerular cells in the afferent arteriole. The stimuli include changes in renal perfusion, which would be detected by the local baroreceptors and the arterial mechanoreceptors. Changes in sodium and chloride at the distal convoluted tubule, which would be detected by the macula densa, which is that collection of cells at the cell wall of the distal tubule where it contacts the glomerulus, so it can detect all that salt and fluid dynamics. There would be increased beta-adrenergic sympathetic activity. So the neighboring cells of those juxtaglomerular cells would be innervated, and upon stimulation, these cells would be able to uh, stimulate the Raas system. And the fourth would be hormonal factors. This would be anything from, say, atrial natriuretic peptide, which would inhibit renin secretion, to some of the feedbacks like angiotensin 2's negative feedback loop with renin, or the inverse relationship that serum potassium has with renin, say hypokalemia causing increases in renin and vice versa. And looking to the organs, angiotensinogen, which is made in the liver, would be released. And with limited utility, it's really only just a precursor to the angiotensin 1. The renin from the kidney, which would be secreted as a result of one of the four stimuli aforementioned, would go ahead and cleave the angiotensinogen on the N-terminal at positions 10 and 11, creating angiotensin 1. So that would go and circulate to the lungs, where it would be converted by angiotensin converting enzyme via cleavage at its histidine and leucine on the C-terminus to give angiotensin 2. Which if you go down here, we could see some cleavage at the histidine and leucine by ACE to create angiotensin 2, after which angiotensin 2 will travel to the adrenal cortex and stimulate the release of aldosterone. So aldosterone is interesting. It's a mineralocorticoid. It's a steroid, so it's hydrophobic. So notice, of course, it's got that the dominant hydrocarbon rings, which are all nonpolar, uncharged, and not very favorable for hydrogen bonding. So it's not going to travel through serum or plasma very well. So it will require a transporter. So typically this will be something like albumin, and albumin will bind to the steroid, the aldosterone, and bring it to the distal tubule cells via the plasma. And we can see here we have aldosterone. We've made it to the distal tubule cell, and made it all the way to the basolateral membrane. So that complex would break down, and the aldosterone would cross the basolateral membrane, then bind to something called a mineralocorticoid receptor before being transported or shuttled to the nucleus. And for the sake of simplicity, I've just used this ubiquitous protein transcription symbol here just to symbolize the effects. Essentially what happens is aldosterone will enter the cytosol there, bind, and upregulate the transcription of epithelial sodium channels, which you can see here on the apical membrane. Leading to the luminal space, as well as it would further activate our sodium potassium adenosine triphosphatase at the basolateral membrane. So this would facilitate sodium transport into the extracellular space and also increase potassium intake, or uptake, sorry, at the apical membrane, while promoting sodium reabsorption. All this to say Simply, aldosterone encourages the reabsorption of sodium and decreases water loss to maintain and increase blood pressure.